So when colonies grow in a lab, the area where the initial cell started the colony is going to determine what type of nutrients the cells have. The cell that started the colony is typically going to be found in that innermost region, and then the cells replicate and move outwards. So the inner region is going to have the majority of the cells, which means there's going to be less nutrients and less oxygen for the bacteria. This is going to lead to death of cells in the middle area, production of waste products that could potentially be toxic, causing increased death. But then the death of the cells actually will provide more nutrients for the bacteria on the outer region of the colony. So the bacteria in the outer region are going to be able to grow and thrive much more effectively. So when we are growing bacteria, we have to look at different environmental factors that allow bacteria to grow most effectively. So remember that prokaryotes can live in almost all environments. The archaea typically are our most extreme bacteria. Most of the bacteria that all of the bacteria that we will deal with in general are going to be able to survive in habitats that humans can survive in as well. The conditions that influence growth are temperature, so cold temperatures versus very hot temperatures, and then the moderate temperatures. Atmosphere, so oxygen requirements for the cells. The pH, acidic versus alkaline, and then the amount of water, the osmotic conditions. Temperature requirements. We have bacteria that can grow most effectively in very cold environments. So these would be found in polar ice caps. Psychrophiles grow at very cold temperatures, whereas psychrotrophs can grow at cold temperatures, but their most effective growth is going to be at around 20 to 30 degrees Celsius. Since they can grow at colder temperatures, these typically are the bacteria that are going to cause food spoilage when they're growing. Mesophiles then are the organisms that grow best between 25 and 45 degrees Celsius. That's their range. And they grow most effectively between 35 and 40 degrees Celsius, which happens to be human body temperature or close around there. So our temperature is 37 degrees, which means these tend to be our pathogens. And then we have the thermophiles. Thermophiles grow at very hot temperatures. So 45 to 70 degree temperatures. These are the organisms that can live in places like hot springs. Hyperthermophiles, 70 to 110 degrees Celsius. These are the severely hot bacteria, or like the really hot environments. Thermophiles then are going to be able to protect themselves because they have proteins that help maintain the integrity of their cell structure. A lot of times we use temperature to protect us from spoilage. So think about the refrigerator keeps our food at around 5 to 10 degrees Celsius. We oftentimes will we will oftentimes freeze foods, and that preserves foods for even longer. What cooling environments do is they inhibit the growth of bacteria or slow the growth, but they don't kill bacteria. high temperatures can kill bacteria depending, unless they're those thermophiles or hyperthermophiles, then they're going to grow well. Temperatures, or the temperatures in different parts of the body can influence the ability of certain bacteria to grow. So our body has a typical temperature or a base temperature of about 37 degrees Celsius. Our ears, fingers, toes, those areas are going to be the coolest of all of the regions of our body. And so some bacteria that grow more effectively in those cooler environments are going to likely grow in those environments, and they can actually cause disease. And one example is leprosy, that causes appendages to fall off, basically. And it causes the appendages to fall off because leprosy grows effectively at cooler body temperatures. Bacteria have to have a certain amount of oxygen, oxygen to survive. So oxygen is needed by all animals and by many of the bacterial species that we talk about. There are different oxygen requirements depending on the species. What I always like to do is I like to explain that oxygen is actually a very deadly gas. And so those original cyanobacteria that started producing oxygen in the environment actually were producing a toxic gas that caused problems for other species. So this is a video. It's, I think, about 5 or 6 minutes. I just think it's really interesting. Oh, I lied. It's only 4 minutes. Or 3 minutes. 2 minutes, whatever.
Hey, wanna play some video games? I know I do. I haven't seen the sun in years. So let's play
Yeah.
I love being outside, the fresh air, that oxygen, that healthy, beautiful, wonderful oxygen.
What I do want to say, I don't think I've used one of these yet, but I love SciShow. So this is Hank Green. He's from SciShow. He's also from the, I don't know, he does another thing on biology, like, I don't remember what it's called, but it helps a lot of students with understanding basic biological concepts. So this is called SciShow, and they talk about a lot of different concepts that people are interested in. And I've always enjoyed it. So since it was brought to my attention, I'll say, in 2012, I think, I've been watching him. Well, yeah, and I like his videos, so you'll probably see him a few times throughout the semester. So enjoy. I'm going to start it over so you can watch it from the beginning.
Love being outside, fresh air, that oxygen, that healthy, beautiful, wonderful oxygen. Don't you love oxygen? Me too. But it's killing us. Today, Earth's atmosphere is only about 20% oxygen. Most of the air that we breathe is nitrogen, with a couple of other trace gases thrown in. But every last member of the kingdom Animalian needs that oxygen to survive because it's how we power our cells. When we breathe in, our blood takes oxygen out of the air through our lungs and carries it to our cells, where it's used in the process of cellular respiration, which is how we turn the food that we eat into the energy that our cells need to do their business. But oxygen wasn't always in the Earth's atmosphere. In fact, oxygen's probably been around for less than half of the Earth's illustrious career, about 2.2 billion years. And it's only been in the past 600 million years that the atmosphere has had enough oxygen to support animal life. So the very first living things on Earth from which all organisms, including us, descended, didn't need oxygen. Which means that we have to deal with something that our most ancient ancestors didn't. Oxygen's dangerous side. Maybe you're familiar with rust, or that brown color your apple turns after you slice it. That is oxygen, reacting with the chemicals in the exposed metal or apple flesh and breaking them down through a process called oxidation. See, oxygen is a needy little element. It's one of those elements that follows the octet rule. It wants to have 8 electrons in its outermost shell, but each atom of oxygen has only 6. And since it needs to be more, it really likes combining with electron-donating atoms. hydrogen when it gets the chance. And that tendency usually serves us well. Oxygen-hydrogen bonding is exactly what allows us to turn sugar into energy during respiration. But sometimes scientists think about 2% of the time oxygen comes out of the process not totally satisfied. So instead of being its normal, mellow, and stable self, it becomes a totally rogue character called a free radical. This thing will bond with practically anything that moves, if you hear what I'm saying. It'll try to pair up with the fats in your cell membranes, or the proteins in your red blood cells, even your DNA, and when it does this, the free radical changes the chemical structure of those molecules, sometimes damaging them. When a protein on a red blood cell is oxidized by a free radical, for example, the protein changes shape and notifies the immune system to take it down in a kind of mercy killing. When cell membranes oxidize, they can become less permeable, and the oxidation of DNA has been known to cause mutations that can lead to some kinds of cancer. Basically, oxygen rusts you out from the inside. This constant cellular wear and tear from free radicals is called oxidative stress, and some scientists think that it's an important factor in aging. Free radicals aren't all bad, though. Our white blood cells actually use them to kill pathogens, and a growing body of evidence shows that free radicals are involved in the signaling between cells. So, while our bodies make the best of the situation, they also try to keep free radical damage to a minimum. That's why we've evolved ways to combat oxidative stress by using antioxidants, or molecules that inhibit oxidation. A lot of foods contain antioxidants like vitamin E and C and beta-carotene, which help protect our body from free radicals by neutralizing them, giving them something to bond to so that they won't have to rip your cells apart. Oxygen. You can't live with it? Actually, you can live with it. You have to live with it. You'll do a pretty good job living with it. I would not suggest trying to live without it. Thanks for watching this SciShow Dose. If you want to continue getting smarter with us, you can go to youtube.com/.
Nope, sorry. And let me go back. There we go. All right, so let's look at the different oxygen requirements that our bacteria may need. We use typically when you're trying to determine oxygen requirements, or if you're trying to figure out what your bacterial cells' oxygen requirements are, you use an agar, well, kind of an agar, that's called fluid thioglycolate. When you boil this fluid thioglycolate, it actually gets rid of the oxygen. And then the bacteria, when you inoculate bacteria, they will grow in different environments. So bacteria that grow at the top of the agar, these are going to be your obligate aerobic bacteria. Obligate aerobes need oxygen. They cannot survive without them. So if we're not bacteria, but if we were, we would be obligate aerobes. We are all animals are obligate aerobes. We need oxygen or we will die. So we're able to produce some superoxides as well as catalase. We produce hydrogen peroxide in our peroxisomes that protect us from oxygen. Facultative anaerobes are organisms that grow with or without oxygen, but they prefer oxygen and grow best with oxygen. So they can grow all throughout the agar, but if you notice, they grow best up here. So they will use oxygen if oxygen is available, but if they don't, then they'll grow without oxygen. An obligate anaerobe will grow only at the bottom of the agar tube. And the reason for this is they can't have oxygen. They don't have those enzymes to protect them from oxygen's rogue and activities. So they grow at this lower environment and do not interact with oxygen at all. I'm going to skip over the microaerophiles and go to the aerotolerant anaerobes. Aerotolerant anaerobes are able to grow in the presence of oxygen, but they do not use oxygen at all. They do produce an enzyme that protects them from oxygen, but they don't use oxygen. So they grow equally throughout the entire agar tube. They'll never use oxygen. Microaerophiles are organisms that use oxygen, but at a smaller amount. So they're going to grow below the top just by a little bit. So they need oxygen, but they need a smaller amount of oxygen and don't like too much oxygen. They don't have as many enzymes to protect them. The organisms that we're going to be using in our lab are going to be these two. So you won't be working with obligate anaerobes. You can cross this one out. You won't be working with microaerophiles or aerotolerant anaerobes. The only two that we will be working with are these two. For the lecture exam, though, you need to know each of these types of bacteria. Okay. Bacteria also can grow at different pHs. Most of our pathogens grow as neutrophiles, and so they're going to grow close to that neutral pH of 7. There are some bacteria that grow better in acidic environments, and so they're going to be growing below 7, typically below 5, and these are called acidophiles. And then we have some bacteria that grow better in alkaline conditions or at a higher pH. So I always like to look at Helicobacter pylori, so this one right here. He grows in the environment of our stomach, which is a very low pH of about 1 or 2. So he grows really effectively there, can't grow in other areas of our body because he is an acidophile. Microbes, just like all organisms, need water. Water is important to life. Without water, we would die. All life would die. But the amount of water we use is going to depend on the different enzymes in our system. So some microbes can grow with minimal water with a higher salt concentration than other microbes. Thank you, sweetie. Halotolerant bacteria grow at higher salt conditions. Halophiles can grow at very high salt conditions. These are typically like your marine bacteria. Extreme halophiles will grow at such high salt conditions that most life wouldn't be able to survive. The Dead Sea is what I always like to look at for that. The bacteria in our lab that we will be dealing with that are halotolerant are Staphylococcus and Micrococcus. These are halotolerant bacteria, and we will actually use an agar that contains salt that will only grow those halotolerant bacteria.